Hey everybody, Mr. Kobe here. So I want to do a makeup lab for those of you who were not in school. So this is a flask with water and a blue chemical called bromothymol blue added to it. Now bromothymol blue is a acid indicator. So it's blue right now, but in the presence of an acid, it'll change colors. And what I'm about to do is I'm about to use this drinking straw right here, and I'm going to blow my breath like that. I'm going to blow my breath through the drinking straw into the flask. And in my breath is carbon dioxide. Now, carbon dioxide is an acid. So after a little bit of time, the carbon dioxide will build up, which is an acid. It'll build up, and the bromothymol blue will start to turn color. At first, it might turn to like a greenish color. So if it's blue, like right now, that means there's no carbon dioxide really in the flask. If it turns a green color, that means there's a little bit of carbon dioxide. And if it goes yellow, that means there's a lot. So I'm going to start blowing my breath right now. So you can see after about, 15-20 seconds, it's kind of turned to a green color. Let me keep going for a little bit. And let me give it one more good breath. Okay, so you can see the color change. Again, carbon dioxide is an acid, and bromothymol blue will react in the presence of an acid, and so that's why it turned colors. So now, what am I going to do with this bromothymol blue and this carbon dioxide? Well, let me pause the video and I'll show you what's next. Okay, I kind of did a lot off camera, so let me show you. Here's the flask that had the bromothymol blue plus my breath that I just blew the bubbles in. And I poured a lot of the bromothymol blue solution into these 4 tubes right here. Now inside tube number 1, there's bromothymol blue, carbon dioxide from my breath. There's a water plant, an aquatic plant by the name of Elodea. You can see it kind of floating in there. And for the next 24 hours, I'm not going to expose it to sunlight. Now, right now, the initial color of the solution in there is yellow-green. We'll see what it looks like 24 hours from now. Tube number 2 has bromothymol blue inside, carbon dioxide from my breath. You can see there's also a watery plant, the Elodea plant. But this one I'm going to expose for the 24 hours to sunlight. Right now, you can see there's a yellow-green color on the inside. We'll see what it looks like 24 hours from now. Tube number 3 has bromothymol blue inside, carbon dioxide from my breath, no Elodea, and this one I'm going to keep in darkness for the next 24 hours. I'm not going to give it any sunlight. And you can see again the initial color is yellow-green. Tube number 4 has bromothymol blue inside, carbon dioxide from my breath. There's no Elodea, but this one I will expose to sunlight for the next 24 hours. Again, right now, you can see the initial color is yellow-green. We'll see if there's any color change after 24 hours. So I'm going to go ahead and put the caps on, and the ones that are going to be in darkness, I'm going to store them in a cabinet, and the ones that are going to be in sunlight, I'll show you them in a moment. Okay, so here are tubes 2 and 4. Not only are they right next to a window, So any sunlight that comes in, they'll be exposed to. But I also have them growing or exposed. I also have them exposed to these two grow lamps here, these plant lights. So you can see tube number 2 has the water plant Elodea in it. Tube number 4 does not. And I'm going to let them sit for 24 hours. Now, in one of my cupboards right here, I have tubes 1 and 3. So again, tube 1 has the Elodea plant, but will not receive sunlight. Tube 3 has the Elodea plant, but will not, or I'm sorry, tube 3 has no plant, and it will not receive sunlight. So I'm going to close the cupboards right there. So I'll come back in 24 hours and show you the results. Okay, so welcome. It is 24 hours later. The next day has passed, or 24 hours has passed. Let me show you the results. I've already gathered the tubes out of where they were stored. So notice the initial color of tube number 1 was a yellow-green, and it's still, the color inside is still kind of a yellowish green color. So no color change happened in tube number 1. What about tube number 2? Notice tube number 2 was initially a yellowish green color, but the liquid inside has clearly gone back to being blue in color. So a pretty substantial color change has happened. Hopefully you'll be able to explain why that occurred. Tube number 3 initially was a yellow-green color on the inside, and it is still a yellow-green color on the inside. So no real color change in tube number 3. And then tube number 4, again, no real color change. Tube number 4 started out as a yellow-green color, and you can see it is still a yellowish-green color. So these are the 4 tubes right here. Based on your lab handout, you should be able to complete the analysis questions. I'll be here if you have any questions for help. Feel free and come talk to me. Hope this was helpful. Thanks.